Hi, everyone, and welcome. My name is Brooke Christensen, and I'm a PhD student in Dr. Monica Daly's lab at the University of California, Irvine. Today, I'm happy to welcome you to another in a series of workshops on integrative organismal modeling of movement. These workshops have been sponsored by the NSF Division of Organismal Systems and will be recorded and shared as an online resource hosted through UCI Center for Integrative Movement Sciences. And with that, thank you all for your attention. And I want to welcome today's first workshop speaker, Natasha Mahatre. Thank you.
Okay, so my talk today is going to be a little bit about, spherical cows and why they're not necessarily the best way of doing things. Or at least they are very, very good in many circumstances. And there are occasional circumstances when you don't want a spherical cow, you want to deflate it and bring it down to its normal shape. So I'll jump right into it and tell you a little bit about what I study. So one of the things that I study is animal communication. I study how different arthropods, insects, and spiders communicate with each other using sound and vibration. And I'm showing you a picture here of two tree crickets. The male is the animal on the bottom, and he uses those wings that he's flipped up over there to make a sound. And that sound then attracts the female, who is at this time mating with him. And the little blob that you see here is spermatheca attached to her, and it's transferring sperm into her. There's a lot more interesting stuff going on in this image, but I'll switch over to talking about how they sing. So how the males sing looks a little bit like this. So the males flip those beautiful forewings up and they rub them against each other, and then the forewing vibrates, and that vibration makes sound. So just a little bit more detail on how that vibration works. So as I said, the forewings are rubbed together. If you take a section through the wing about there, and you look on the underside of the wing that's on the top, you see these pegs. And what's happening is the edge of the lower wing is catching each of these pegs, and every time it catches and releases the peg, it sets this wing, the whole plate of the wing, into vibration. So you get basically what looks like an out-of-plane vibration, and that is what makes the sound that you hear. So here is just a reconstruction from laser vibrometry data of what that motion might look like. So the wing vibrates out of its plane, and that is the thing that generates the sound that you hear. And one of the things that you might want to know is how much of a vibration makes how much of a sound. What does that sound look like? And that's the kind of thing people like me are interested in for a lot of different reasons. And how do you go about answering a question like that? Like, when something vibrates, how much sound is it going to make? And the sort of classic way of doing this was to use analytical models of vibration. So this is, a review I give to every single one of my students that are working in acoustics, and say, read it, because it has just absolutely the best density of information for starting to get into this. And essentially what this review kind of tries to do is explain how structural vibrations become acoustic sound fields. And it does it extremely well. It explains everything really, really well. But when you look very deeply into what it's using in terms of analytical models to explain how sound is produced, is it uses very, very idealized sort of setups of sound and vibration. So you can write a really, really nice, clean equation for how much sound something like a baffled piston makes. So a baffled piston is simply a piston which vibrates up and down, like this piston over here is, and is sort of surrounded by this object, which we call a baffle. It's the thing that allows the pressure in the front of the piston to be separated from the pressure in the back of the piston. And you can write a really beautiful analytical solution to this, which will tell you exactly what the sound field will look like. And, if you have a ratio of the size of the piston to the wavelength of the sound that it's producing, you can get these really nicely parameterized pictures of the sound field. And it's you can go a long way with this in terms of trying to understand what sounds animals make. So for instance, I'll just show you in a minute a whole series of really good experiments that used exactly this model, a baffled piston model, to make really, really interesting observations about animals out there. But one thing to think about over here is this is a spherical cow. This is a spherical cow because this is a perfectly circular piston in an infinite baffle. So that's also something that is assumed in this analytical solution, that this baffle is infinitely large. Now, a cricket is not sitting in an infinite baffle. Its wings aren't perfectly circular, and is no simple way to, you can kind of look at this system in analytical terms, but basically I'd have to write out the math to do that, or an engineer would have to. And that makes things really, really difficult. And I'll get into that in a little bit. But people have gone a long way with these kinds of really simple piston in an infinite baffle or a vibrating sphere model, and they can kind of use that to make really intelligent and educated guesses about how loud lots of different animals, like a huge range of animals, can be. And you can even make very, very specific predictions about, say, for instance, how loud a bat will be and what its sonar beam will be. You can get really, good sort of expectations which you can verify against data, and you can get a paper into Nature, for instance, using this kind of model. The problem becomes when you want to actually ask questions about some of the things that don't quite meet the assumptions that kind of spherical cow forces you to take. So for instance, I want to know how much sound the wing of this cricket will produce. I want to know how much sound the wing of this cricket will produce when it's on its own and when it's in a baffle. And the baffle that I'm talking about is not infinite. It's a very real leaf. It's a leaf that has a particular shape and a particular size. And that's what I care about. So that becomes really difficult when trying to use that kind of analytical equation. And then our decision was that the best thing that we can use is something like a finite element model. So finite element methods, really, if you're a mathematician, people don't like calling it a model. They prefer to say, did you set up the problem or you set up the model, then you use the finite element method to solve it. But essentially what it can do, it numerically solves equations for a particular geometry. So using the finite element method, and I won't go into the details of it, you can, for instance, ask what the sound field radiating from a simple vibrating piston can be. You can also ask what it can be when it's vibrating in a baffle. You can change the shape of the piston, you can change the shape of the baffle, you can use different wavelengths of sound, and you can get really good answers for what kind of sound field will come out of this. And this is why we kind of decided we want to know things when they're unbaffled. We want to know when they're in imperfect baffles. We want to know what the sound field is going to look like when they're baffles of different size. And you can't do that with an analytical model. So we decided that if we wanted to study tool use in these guys, tree crickets, we needed something that was more flexible in terms of the geometry of the problem. And that's sort of when we decided that we were going to use FE. So I'm just going to kind of show you some of the data that you've already seen. If you've read this paper. So what we did was we convinced 5 tree crickets to sing from a small baffle that we made for them. They sang, they were nice to us. We measured how much their wings vibrated. So here's just some data on how much their wings vibrated. They vibrated about 2 meters per second at peak velocity, and that produced about 108 millipascals of sound at peak sound pressure, which sort of was 74 dB at peak, but when you averaged over time, it was a little bit less, as you can see from the structure of the sound itself. And what we did next was to make an FE model out of this. So that's what the averaged values look like. We made an FE model out of this that kind of tried to respect some things about the geometry of the system we were looking at. So we had our animal suspended in free air, sorry, and there's our cricket in its little baffle in there. So that's, you can see you don't have to model every single piece of the geometry absolutely perfectly. This will give you a pretty good prediction. That's not a cricket with all its legs or all its heads, but it's good enough for what we're trying to do. And in fact, you can even lose the body and it doesn't really make much of a difference. And then that can predict the sound field that comes out of this kind of animal and this kind of baffle. And we've made measurements from actual crickets singing from actual baffles at a particular distance. So we have point measurements and we can compare them that to our prediction. And they're a pretty good match. Basically, we get a measurement of about 60 dB and we get a prediction of about 61.9 dB, which is really within sort of what the expected limits would be. So a model like this is pretty good at predicting both what the sound field looks like and what the actual values of that sound field are. And you can now check these against, to an extent, against expectations from analytical models, and they seem to be okay. So you can move on from there and do a lot more than you're able to do in the analytical models. So for instance, we're interested in tool use, right? So we want to know, like, what happens when the leaf is a small leaf? What happens when the leaf is a big leaf? Because we know a small leaf is going to be worse at baffling than a big leaf is going to be. But exactly how different are they going to be? What happens when the hole is the same size as the animal? What happens when it's a little bit bigger? Because we know same size is going to work better than one that's a little bit bigger. So those are the questions you can start to answer, which you couldn't with the other system. Now, here's another sort of, thing that I wanted to show you. So I sat yesterday and dug through, I don't know, last 7 years of data to try and find this piece of data that we discarded somewhere along the line to publishing this work. So when we began this exercise, we didn't make our tools or our baffles look like the leaves. What we did when we began was to just say, well, we can idealize this down to just a simple oval, right? It doesn't matter. A leaf is a leaf. I mean, a baffle is a baffle. As long as it looks roughly right, it has the same width and the same height, it'll work just fine, right? So that's how we started with this. And we sort of modeled different size baffles. So the leaf was different size, the hole was different size. Remember, we had two geometries, either the oval one or the more realistic one, where you can kind of see the actual shape of the baffle. And we had sort of these efficiency maps for both of them. So the one on the left is something we never published, which is from an idealized sort of baffle. And what you have on the x-axis here is sort of the oval of different heights. And on the y-axis is the oval of the whole oval of different heights. And what we say is, for these combinations of parameters, what is the efficiency of that baffle? So if the wing vibrates this much, how much sound does it produce? So per unit vibration, how much sound is produced? You can see that, okay, there's an optimum point, and we did a bunch of experiments where we offered the animals two sets of leaves, and they always chose to baffle from the larger leaf, and those are the kinds of positions that they sit on in this efficiency map. And what's on the right is the data that we actually published. So this was data where we changed the baffle that we used into a leaf-sized, leaf-shaped baffle. And you can see that the predictions from the two are actually different. Although the heights are similar, whether we're talking about the leaf height or the whole height, they're pretty similar, but the predictions are are different in detail and also in overall efficiency that is predicted. So we tend to see higher efficiencies when we respect the actual shape of the baffle than when we just call it an oval. Also, as it turned out, luckily for us, we picked leaves of the right size, whereas this told us that we hadn't picked leaves of the right size when we kind of did our experiment to test whether the animals were picking the best, most efficient tool possible to make. And this looks bad, this looks good. Basically, is the sum of the story. And the only way it would look this good is if you actually respected the geometry of the system and didn't treat it like a spherical cow. We could also do other things with this, which we couldn't with sort of an analytical model. We could worry about the fact that animals are animals and they do bizarre things. For instance, instead of putting the hole in the center where it's best to, they put it off center sometimes, and we can ask questions about, well, when they go off center, how much do they lose by going off center? And again, you kind of have an answer to that over here from a finite element model. You couldn't get it from an analytical. I keep harping on analytical models. They're not terrible, but you can get like these really detailed pieces of information, and you can kind of say things like, oh, the drill baffles that they made were about 90% as efficient as the best possible things they could make. And that's, that's what a biologist is interested in quite often. So, and that's why I think these are really useful kinds of ways to think about things. So I'm going to give you another little taste of what we do with these kinds of models. So how we use them to deal with even more diverse sort of forms that animals take, right? Because that's what biologists are interested in: diversity. They're interested in complexity. So one of the questions that turned up when we finished that paper was like, well, um, if baffling is so good, why don't all the crickets out there in the world baffle? And we can start to ask this question by thinking about all of the different shapes that crickets take and all of the different places that they call from, and ask, well, if we put these guys in a baffle, how much better would their calls be? And maybe you will find out that it's not that much better, and that's why they don't all baffle. But again, now you have to deal with the fact that cricket wings are not simple circular pistons. You have to deal with the fact that they call from weird things like from the ground or from the vegetation or from leaves. And, given all of this complexity, what is really the utility, or what is the efficiency gains you can get from baffles? And there really is a lot of diversity. There's close to about 6,000 different species of crickets out there, and they really vary in size, they vary in the shape of their wings. Can you actually even begin to answer something like that? And this is where we've started using FE, and I think in the best possible sort of way that we can. So when I say we, it's the really, really royal we. All of this work was not done by me, but was done by my mentees in the lab, Aaron Brandt, who's a postdoc, and a whole slew of undergrads who really, really helped us out. And what they did was to go out there and collect data on the shapes and sizes of different wings and their frequency, the frequency that this wing was made to radiate. And we did this we tried to be respectful of the phylogeny of crickets. So that's the number of species in the known clades right now. And that's the number of species that we sampled. And we tried to make sure that we weren't oversampling any of the clades. We tried to spread it out. I think we did a pretty good job there. And then we asked, well, if you took all of these guys and you looked at when their wings vibrated, how much sound they were able to make in terms of efficiency, what would that look like? So these are all the crickets we have. These are the sizes of their wings. These are the frequencies that they sing at. And it turns out we did a little bit of the work. It doesn't really matter so much exactly what shape the wing is, but you have to get the surface area right to make a good prediction of how much sound it's going to radiate. And here is a kind of landscape of predictions for how efficient these wings are going to be. And one thing that jumps out at you immediately is every cricket in the world, pretty much every cricket in the world of all these 100 species that we sampled, is really not as efficient as it could be. So this sort of ridge, we call the ridge of high efficiency, is where they should be if they were optimally efficient or equivalent to being in an optimal baffle. Nobody is. So every one of these animals could scooch over to the right of this by using a baffle and could become more efficient. They really, really had a lot to gain. So the question is, why not? And here you have to start to worry about the fact that everything we're doing is in highly idealized conditions. So we were, in our baffling paper before, it was reasonable to put an animal in a baffle in free space, but for real animals, things are a bit more complex. Real animals don't baffle in free space. They're kind of screaming from the middle of vegetation. They might be at the edge of the vegetation, but they still may have to deal with reflections from the ground. Some of them call from the ground. What does that do to everything, to their sound field? And here we found that it wasn't enough to use FE as a method to deal with complexity. We had to move over to a new kind of model, which is computationally less expensive. This is called boundary element models. And we decided to do that. And the cool thing about this then is you can sort of have a radiator that's making sound, and then you can add in different pieces of information. So one of the pieces of information that you can add in is that you can say, well, there's a ground here, and that ground has a particular impedance, and it's this reflective and this dissipative. And when can you please tell me what the sound field will look like when you do that? You can add in information like, oh, this is the kind of excess attenuation you get when there's these kinds of crops on the ground, or there's this much attenuation when you have a different kind of vegetation on the ground. And you can use that kind of data along with your BE models to predict how much sound something is going to make. So we start to make models like this, and we had to cut down the space of our problem. We had to say goodbye to these animals, and we were kind of happy to, because they're actually a bit strange. They use higher modes to sing, so we were kind of happy not to use those in our modeling, and we limited ourselves to like a smaller area of the state space that was available and did boundary element models. And we were happy to see, when we were sort of talking about things in free space, the predictions of both of these match relatively well. But what was different about both of these is, in the FE models, we could only really model an acoustic field of about 20 centimeters. In this sort of system, we can model an acoustic field that goes as far as 1 meter away from the singer. And that, really gives you a lot of power when thinking about not just how loud an animal is, but how loud it is when its sound propagates through space. Because 1 meter is kind of the right range over which animals are heard by their conspecific mates. So now you can ask that question. What happens when an animal is calling next to the ground? What happens when it's calling way up in the air or from vegetation? And there's a lot of results, and we're releasing our preprint really soon, so look out for that. But I'll kind of cut to the chase. I'll cut to the interesting part, which is that it turned out that actually baffling was great, but very close second to baffling was calling from the ground. So it turned out that the ground acted as a baffle and really allowed animals to be quite efficient. So this is when the animals are in free air, and you'll notice that the highest efficiencies here are significantly lower than the highest efficiencies near the ground. So that's what this data is. And this is really cool because it suggests that we've been thinking about this in the wrong way. We shouldn't ask the question of why don't all animals baffle, but we should ask the question of why did animals leave the ground at all? Because the ground is such an efficient place to call from. And essentially what is happening is the ground acts as a baffle. So what sound would normally be lost to the air over here is reflected from the ground and then radiated upwards, and you have actually quite a lot of opportunity for constructive interference, and you can kind of get really loud sound out of it. And being biologists that we are, we put it back all into the animals. We looked at, all of those animals in those clades I talked about earlier. If they called from the ground, how efficient would they be? If they called from the vegetation, from a baffle, how efficient would they be? If they didn't have a baffle, how efficient would they be? And it turns out for at least some groups of animals, actually calling from the ground is almost as efficient, if not more efficient, than calling from a baffle. And it turns out that you can, when you think about it in these kind of more complex ways, which are not possible in the analytical system, you can have really cool biological insights, which is that actually calling from the ground is a really, really close alternative second to calling from a baffle. And maybe that's why we don't see tool use across the whole cricket phylogeny. All right, that's it from me. And I'll stop there, and I will acknowledge all of the people I've done this work with and my funders. So I'm going to stop, hand it over to whoever, and mute myself.
All right, so my presentation is not a modeling one per se. This is really more technique based. So this is about a fancy form of imaging called synchrotron X-ray imaging. And from this, you can actually develop lots of different models, and we have as well. But I actually wanted to focus on the technique because I think, you know, we've been using this for quite a while, but I'm not sure how widely known or appreciated it is as something that people can use. So today I'm going to give you a brief history. I'm going to talk to you about what it is and how does it work. And then the three main things I'm going to talk about, I'm going to give you some examples from 2D imaging, which is really full field imaging. And I'm going to talk about contrast enhancement. And then lastly, I'll talk about tomography. Okay, so brief history of how we got into this. So Wah-Kat Lee is a physicist. He worked at the Advanced Photon Source of Argonne National Lab, which I'll show you quite a bit in a few minutes. And he's not a biologist. And he went outside and collected an ant and stuck it in his imaging device and collected this X-ray. So you're looking at that very X-ray. And not being sure what to do with it, he looked around the area and said, oh, the Field Museum has lots of biologists, and sent that image off to the Field Museum. The person who got interested in this was Mark Westneat, who, if you know him, works on fishes. But Mark was keen enough to go, this is really cool. And from that, he started looking at all kinds of insects in the beam. And one of the first things that it led to was this cover paper in Science, where you see a beetle here that is compressing its tracheal system in a rhythmic fashion. Just to give you a sense of scale, and this is really relevant to the technique, the vertical axis here, the y-axis is about 2 millimeters, and the x-axis is about 3 millimeters. And at the main beam line where we do this work, that's pretty much the limit of what you can see with this particular technique. So currently it's about 3 by 3 millimeters. There are other places around the world where you can go bigger, but not at this facility. Okay, so my talk today is really about how you get this type of image, what's involved. So to back up a little bit, so these things are called synchrotron light sources, and they're not unique to the United States. In fact, they're found in many places around the world, and there are about 50 that are in operation or under construction. This website is a really good source to learn about this type of thing, lightsources.org, for future reference there. And so what are these things? So first of all, when you look at them, they're these giant facilities. They tend to be in rings, and that's for a reason. And I'll go into the physics of that in just a minute. But you can see they're various sizes and shapes. And the one that I've done the most work on is from the Advanced Photon Source. And again, I'll show you that one in a second. So what type of work gets done there? So if you look at this pie chart here, you see that it's all kinds of work, with the majority of it being biological or life sciences. It's a little bit deceiving. So I'm an organismal biologist and a biomechanist, and my type of work really is not represented. You know, this mostly is, you know, there's a lot of protein structures that are, or proteins that are not crystallizable, whose structure is determined at synchrotron light sources. So they've been really invaluable there, and that's where the bulk of this work is. Okay, so let's get into how this actually works. So to start with, X-rays are a form of light. So when we say that these are a light source, it's because X-rays are electromagnetic radiation, where the energy of the photon is equal to Planck's constant times the frequency. And that's also equal to the Planck's constant times the speed of light divided by the wavelength of that photon. And so you can then think of the photon's energy as being inversely proportional to its wavelength. Typically, when people talk about synchrotrons, they're talking about the energy, but if you want to think about it in terms of wavelength, you're welcome to do that as well. And typically at these facilities, they're dealing with X-ray energy sort of roughly in the range of 1 to 100 keV or so. Okay, so how does this work? So to start out with, light interacts with matter in a number of different ways. You can get reflection, refraction, where you see this, looks like the light is bent here through this different medium. Of course, you have absorption of light. And then lastly, there's diffraction. And all of these things, because X-rays are light, all of these things are possible with X-rays as well. And the imaging that we use takes advantage of some of these features, particularly of diffraction, refraction, and absorption. Okay, so when an X-ray passes through matter, so it can either be scattered or transmitted or absorbed. And we actually don't deal with, in the full field imaging, we don't deal with scattering. Okay, so scattering types of experiments that you might be more familiar with, of course, the classic DNA experiments. That's not what we're doing here. So we are actually dealing with full field images, and so that works because you have the photons either pass right through or they are absorbed by the matter. Now, what's a synchrotron X-ray? Well, the bottom line here is a synchrotron X-ray is a form of X-ray that's produced at a synchrotron. So let me show you what a synchrotron is. So essentially what you have is an E gun. That it means that it's an electron gun. And for those people in the audience who remember large TVs, so in your old style TV, what was in there was an electron gun. An electron gun sprayed electrons onto the screen, and that's how you form the image. Here, it's essentially the same thing. You form these electrons, but the difference here is they get boosted in energy up to relativistic speeds. So these are electrons traveling very, very fast. They're then injected into a storage ring where they travel in straight sections, and then they're used either in an insertion device or a bending magnet device. And from these, you get these tangential beam lines that come off the facility. So this is just a schematic. Let me show you a picture of a real synchrotron, and I'll show you where all the beam lines are as well. So this is the advanced photon source. It was built in the late 90s, and it is at Argonne National Lab, which is 42 miles west of Chicago. And the reason why it is kind of outside of Chicago is that this lab was started by Fermi. Fermi built the first atomic pile at the University of Chicago in the squash court. They realized that this was a bad place to potentially blow up an atomic bomb. And so they moved the facility out to the prairie at Argonne here, which is now Lamont. So we do our work particularly at beamline 32ID, as well as 2BM. And recently, there's been imaging that's started up at beamline 7 as well, and I haven't worked there, so I don't know much about it. But these are the three places where you have imaging at the advanced photon source. So the vast majority of this facility is not devoted to imaging. Okay, so This is a more schematic view of the APS. So in the blue, there is the storage ring, and in the purple are individual beam lines. So you see those all come off in tangents. And like I said previously, the majority of these are not for imaging. Okay. Now the basis of how the X-rays are produced, I'll go into that in a second, but essentially you have magnets, and the way that what happens is your electrons, which are traveling at relativistic speeds, when they pass through a vertical magnetic field, they are bent. And when they are bent and accelerated, they produce a radiation. Okay, and it is that radiation that is used usefully. Now, an important quality of these X-rays is that they are partially coherent. Okay, and they're coherent both in time and space. They're not perfect like they're like depicted down here. If you look at a laser, lasers are more coherent than the X-rays that are produced here, but the partial coherence enables you to take advantage of the previous effects that we talked about. View from the inside. So what you're looking at here is both the storage ring and the booster ring. This is not from the APS, but it looks very similar. So these are giant facilities. When you walk into them, it's incredibly impressive. And even if you're working on things like a bug, like I am, you're like, wow, this feels like science. So it's fun to do. Okay, so bending magnet beam lines. So how they work is it's a giant magnet, and here the magnetic field is coming out of the plane of the slide here, and so the electrons are passing through. The electrons are accelerated, so their direction is bent, and then you have this spray of radiation that comes out here tangentially, and as a result, at a bending magnet beam line, you can actually get wide beam. Okay, so I was saying previously that our limit was about 3 millimeters in width. At a bending magnet, you get more width than that, but you still are limited in terms of height of the beam. So at 2 bm, the height limit is about 2 millimeters. So the other type of beam line are the ID beam lines. These are insertion devices. They're either a wiggler or an undulator. Wigglers and undulators are about the same thing. They just differ in how much they wiggle the electron. So wigglers cause a greater deflection than do the undulators. But essentially, in these types of beam lines, you have the same type of thing where you have this, the electrons are passing through a magnetic field, but they are flipped in polarity in sequence. And so what happens is the electron passing through there turns one way, then it turns the other direction, then it turns another one. And every time you do that, you collimate the X-rays. And so what you get as a result of this is an extremely powerful X-ray. Okay, so here we're looking at the photon energy in kiloelectron volts of X-rays at a synchrotron light source. And this is brilliance, which is just the way that the intensity of the beam is truly measured. And what you see is that in ID beam lines, you get these big peaks of brilliance, whereas in the BM beam lines, it's not quite as powerful. Okay, so that's the advantage of the ID beam line. Now for us, in trying to image live insects, we do not use this power. Okay, so if we actually went to one of these peaks, that the animal is dead very, very quickly. And by fast, I mean really, really fast. So we actually tune these down when we are doing our work. Now there's different types of light that can be produced. There's white beam, which has a broader range of wavelengths or energies of the X-ray. But typically we've done our work using a monochromator. What a monochromator is, it is a device that has two crystals, and you're essentially the raw X-ray beam that has a broad bandwidth comes in, and only a very small bandwidth of X-rays is reflected from that crystal. The rest of it passes straight through. Then you have a second crystal to get it going back in the same direction. And so now you have monochromatic light. It's not truly one wavelength. It's still a bandwidth of X-rays that passes through, but it's a much smaller bandwidth. And I'll show you why that can be advantageous later. This is just a slide showing that we measure things in brilliance, and that's the photon flux per area per unit solid angle at the source. Yeah, there you go. Okay, so let's talk about practical stuff. So how do you actually do these experiments? They're done in a steel hutch. And in the steel hutch, you cannot be. Okay, so there's actually safety devices in there. So if you ever get caught in one and the beam is on, there's a safety and you punch that and it shuts down the whole facility. As far as I'm aware, it's never happened at the APS, but they have that capability. So there's the hutch. This is what they look like in practice. So this is 2BM, and note this is B. So there are multiple stations along the way. This is 32ID, and that's me for scale. I'm kind of short, so it's not that this is that big. Okay, so how does this work? So let's talk about phase contrast imaging first. So as I was saying, you have the X-rays come out of a source, and then they pass through the monochromator. You have your monochromatic X-rays. They then pass through the insect, and then the key to this technique is that your visualization of the X-rays occurs at a distance from the insect. And the way it happens is that the X-rays pass through a scintillator. And it's a very thin crystal, and that crystal converts the X-rays to visible light. So on the inside here, you have a visible light image, and that then can be imaged with any sort of imaging device. Okay, so here this is pretty old now. We don't use CCD video cameras anymore, but any type of camera that you want to put on there, you do that. This mirror here is simply so that it's reflecting only the visible light, and we're not frying our instrumentation with X-rays that are passing through. Okay, and so given that you have the partial coherence, what you get is a phase contrast effect with this distance. So if you on the left here is an absorption only image, so like at the scintillator or right against this insect here, and on the right is a phase enhanced image where our insect is in the range of ½ meter to a meter away. Sometimes we work closer as well, but you can see the details that now pop out of this ant head. Okay, so what does it look like in practice? So this is inside the hutch on an optical table. There's a bunch of translational stages and a rotation stage here, and our sample is placed up here. And the reason for this is so that you can control the position precisely from outside of the hutch. And then the X-ray passes through the insect, and then it hits the scintillator screen over here, and I'll show you a picture of that. There's the scintillator. This is an old style system that we used to have where here's the mirror, and it was put on a rotation motor so that we could switch from this camera to that camera quickly. But typically we've stopped using this in the past couple of years. But you can see that this is just a microscope optic, and then an empty tube, and then the camera is behind this piece of metal here that you can't see. So the scintillator was out there recently. They've moved to putting the scintillator and the mirror into an enclosed box here, and then using this Kapton film, which is translucent to the X-rays, but it blocks visible light. And so we can work in the hutch here with you can do your experiments with the lights on. You don't have to turn them off to make it more sensitive. And you can see this is a high-speed camera here connected to this box. So here's an experiment where we actually had two high-speed cameras. So this one was for the X-rays, and this one was to see what the animal is doing with regular visible light. So this just shows you too that you can bring in all kinds of other instrumentation into your system to do experiments there. Now that table was kind of cluttered. This is a picture from very recently. I think it was last week or the week before. I can't remember now. My brain is a little scrambled. But you can see this is a much more clean setup. This camera here is really tiny, but it's very sensitive and produces really high quality images. It's connected to the scintillator box there, and there's our sample there. There's the sample mounted. This is just a cockroach in capped on tube. And I wanted to show you this one too as well, because if we zoom in here, and you can see that the translation and rotation stages here are much smaller because we're not wanting to hold up much weight here, and so it can be more thin. All right, so I want to show you this video now. This is doing an experiment in practice. So here I'm mounting the sample. I don't take much time here of aligning it, but I would. That was just for show. And what you have to do is you have to search the station. So you go in and you press the button at one end, and then an alarm starts going.
Searching station C.
The woman warns you that you need to exit immediately. So you now make sure there's no one else in touch there. And hit the other button, and now you come and close the steel door automatically. That takes a bit. And one of the reasons I'm showing you this is that if you want to do an experiment that involves timing with the animal, you have to think about that carefully. You have about a minute or so before everything is set up, and now you can come to the control station and actually turn on the X-ray beam. Okay, so a little bit more about the imaging itself. So I was talking about different energies of the X-rays. What effect do you have? So as you increase in energy, you actually get less absorption by the animal. So more of those X-rays pass right through. So you can see in this axis, the kind of the image going away. So working at lower energies is better, but there's a trade-off, because as you'll see in a second, that this kind of does more harm to the animal. And then on the y-axis here, we have sample to detector distance. So this is close at 5 centimeters. This is 50 and 100. So you see, as you pull away, you increase your phase effect. So this was an early experiment where we're simply trying to see how long the different insects could withstand being in the beam. And you can see when the beam is on here, this blue is a respiratory signal, and the animal was doing okay for a while. Then you see things start to change, and typically there's a respiratory signal where something happened in each of these different species. And so that's what we're plotting here. So this is the power of the beam. In other words, you get more photons on the animal, and increasingly it makes a better and better image. Okay, so this is a poor image, and this is a nicer one, or this is probably the nicest one, a little blown out. But you can see here that the time to this respiratory signal goes down. That means that the animal's dying more quickly, right? So you want to be working up here, and so you want to lower this as much as possible. Now, the way you actually do this in practice is by changing the gap. So this is the gap control here, and what you're doing is you're changing the distance between the magnets. So as you increase the distance, what happens is the magnetic field gets weaker, and so with the weaker field, you produce fewer X-rays in general. Okay, so that is how things are done for 2D imaging. I'll get to 3D imaging in a second. I just wanted to show you just a couple examples. So this was from a study. So the advantage of this technique is you see things inside that you can't outside, right? So here's our opaque caterpillar, and it's actually on a little treadmill here. And what you can see that's cool is on the inside, we have tracheal tubes that are whooshing by as the animal is moving. So in the purple there are sort of tips of tracheal tubes, and you can see their movement. And what's interesting here is that they're actually they are synchronized with the back end of the animal. The back end of the animal is essentially pushing the gut forward within the animal as like sort of a gut piston. Okay, so something you can only see with X-rays. Okay, so the image I just showed you is just pure X-ray, but you can also add contrast agents. So on the left, we are adding iodine, and on the right, we've added a cadmium agent into the food, and you can see here the gut. So now we see movements of the gut better, because essentially, if you have density, if you lack density, strong density differences, you still don't see things well, even with the phase contrast. So the visualizing the food, we're the way we do that is we mix iodine with the food, and then we tune our X-rays just above the K edge of iodine. And so what that does is causes a local resonance in essence, and you get this big absorption here. And so if we were below this energy, so if we were at say 33.1, this goes away. Okay. So here's an example where we used that technique in mosquitoes. So we had them drink a glucose solution that was mixed with the iodine, and then now you can see drinking inside the mosquito. So you can see it going through its system there. And this is a bit more interesting, where we discovered a mode of drinking where you get this very big bolus that's created in the secondary pump there. So, and again, without that, you can't really see the pumps. Another method that we use is to inject microbubbles. So here's a grasshopper where we're trying to visualize blood flow. And this was an early experiment in about 2007. So the camera quality here, this is essentially our first camera that we used. But you can see bubbles here in the heart, which is pretty neat. And so you can do things like track bubbles. And so there is, here we're going to identify a bubble in yellow. And so you see that now entering into the heart. We've also done work with high-speed cameras here and other microspheres. So these are not bubbles; these are glass microspheres. And this is here slowed down. You can now see oscillation in the heart of this grasshopper, which we haven't published yet. You can also do very, very fast imaging. So this is an example that was published recently. And this was done on click beetles. So I want to show you something. This is a movie that we made in 2005. And you can see the click beetle there. It's clicking madly. And if you freeze any one of the frames, you can see all this blurring here. And that is because this click motion is incredibly fast. It's one of the fastest motions in biology. And now with improvements in our sensors, we can now do things like image at 30,000 frames per second with the X-rays. So this now is a more modern movie where you see the click there. And here now we don't have any kind of motion blur whatsoever. Lastly, I briefly want to mention tomography. So tomography is a technique where you're making 2D images, the same type of projection images, but you're rotating your sample over 180 degrees or 360, and you're using that to reconstruct the image in 3 dimensions. Okay, so in essence, you get a series of slices that you can then, you essentially have 3-dimensional data. So for that type of work, we need the animals typically to be as still as possible. Although people have done live imaging, I don't have a slide here. I would recommend looking up Simon Walker's work, as well as Moscow's follow-up paper. It's really quite incredible. So in this technique, you have this really nice rotational stage here. Here you're seeing 3 sets of scintillators and optical components. And this is now at 2BM here. They're able to change the magnification on the fly, which is really quite nice. And then you get images like this one. This is a slice from a beetle. And then you can reconstruct things like the tracheal system here, which is particularly nice because you can do things like taking a single tube there rotating it so that you get the orthogonal axis. And now we can do things like look at the different cross-sectional areas of the tracheal tubes. Okay, I kind of blew through that tomography stuff pretty quickly. But that's because I want to make sure we have enough time for our other presenters. And I'm happy to take questions later as well, too.
So welcome, everyone. I'm really happy to talk to all of you about robogami modeling. So broadly talking about how do we think about robots and using origami or kirigami techniques to build our robots. And I want to start off by saying a big part of our group's research, in addition to building robots with increasing capabilities, is for us, robots really offer us a really nice way to understand and test hypotheses of biological systems. And we had a great workshop just a few weeks ago with a number of people talking about the utility of physical models. And if you haven't gotten a chance to check that out, please go look them up. They talk about a number of techniques of how to generate physical models, mostly focusing on 3D printing based approaches to replicating the mechanics and the structure of biological organisms. So why do we need? So there are a lot of techniques out there. And in our group, we specifically use a kirigami-based approach to basically generating these physical models. And there are a couple of main reasons why we do that. One is we're trying to build structures or physical models at small scales, approaching the size of insects. So here's, you can see local tarantula next to our quadrupedal robot. And so on the physical modeling side, it's really advantageous to build robots at the same scale as animals, because then we don't need to worry about the physics of scaling. A lot of times we're trying to explore the underlying mechanisms, and if we can build physical models at the same scale, then we don't need to think about scaling. So, and there are ways to accommodate the effects of scaling. For example, like in fluids, we use things like Reynolds number to just match properties and identify interesting principles. But if we can build structures at the same scale, then that's really advantageous, and it makes a lot of the analysis simpler. Another reason we try to do this is, as I'm going to try to explain, we saw wonderful talks today by Natasha and Jake, and some of the techniques they were talking about are, they're not mechanics-based. So Natasha talked a lot about sensing and communication, and Jake talked about trying to understand fluidic structures within the body, and oftentimes with some of the other techniques, it can be quite challenging to incorporate sensing structures or begin to think about how to incorporate fluidic systems within your entire robotic structure. And as I'm going to show you towards the end of this presentation, is this origami-based approach, which is essentially a stacked laminate creation approach, enables us to actually integrate those pretty well. So, so some of the history of this work, in the last 25, 30 years, a lot of the groups have been really interested in making small-scale robots. Here's a picture from Rob Wood's lab, where this was part of a big NSF project, where the idea was to basically be able to create robotic insects similar to the size of flying bees, which could potentially be used to pollinate flowers at some point. And so, so when this project started, there were a lot of challenges in terms of thinking about how do we actually design systems to the scale, what do we do in terms of actuators, sensors, and all of those things. And over the last couple of decades, we've made significant progress, and I'm just going to highlight some work which in this direction. And so one of the things I want to point out is, as we start thinking about these really small structures, it's really one of the challenges is that it becomes really difficult to use the classical nuts and bolts kind of approach, where you can make really small parts, but begin to collect all of them and assemble all of them into monolithic structure becomes really tedious. Like, so for example, if you build a car, there are lots of structures, there are even some really small parts. It takes work to do that. But when we start like shrinking a whole car down to the size of an insect, it's really challenging, and we need really talented graduate students to accomplish some of those tasks. And so one of the approaches, and that was in part what was motivated things to think about interesting alternatives to design. And so one of the ideas was looking at insects in terms of like how they're built. And one of the key features of insects are that they have these like exoskeletal systems, which are essentially rigid plates and tubes interconnected by membranes, flexible membranes, which allow them to not only be tough, but also be compliant in select directions. And this enables the exoskeleton to serve a number of functions, including support, protection, anchoring muscles, and also enabling locomotion sensing, and among other things. And so, as Jake showed you, so here's a so using some of the similar optical techniques. So we did a micro CT of an American cockroach, trying to look at the structures inside. And here is a dissection which I did of Blabris, a slightly larger species of cockroach. And what we observed was essentially the exoskeleton was made of these like plates, which were interconnected by soft tissue layers, which enabled the animal system to basically be really compliant. And so we started taking advantage of this approach and started thinking about if this could be something amenable to translate into robotic structures. At the same time, with collaborators in Ron Fearing's lab at Berkeley had already started thinking about ways to do that. And in fact, one of the approaches they came up with is basically combining stacks of rigid cardboard with the polymer, like polyamide in between, and laminating the whole structure together. And so basically by selectively having cutouts, we simulate places where you can create joints, and by combining rigid material like cardboard along with the flexible material, we get a laminate structure, which can then be used to like fabricate an entire robot. And so this, and so one of the techniques, one of the earliest techniques which was used to kind of use origami-based approaches into this robot modeling structure was to basically was a approach called the smart composite microstructures approach, which I mentioned was kind of looking at partially inspired by looking at animal structures and exoskeletal structures. So in this approach, what we do is we basically start off with a 2D drawing. So basically we try to identify where we want flexible elements and where we want rigid elements. Wherever we need flexible elements, we cut slots using a laser. We insert a polymer and then bond all the layers. Typically, you have a rigid layer, a flexible layer, and a rigid layer. So, and the three of them are bonded with potentially additional adhesive layers in between. And then we again like stick this in a laser and cut out individual parts. And as you see, and then you can fold out those individual parts and assemble them into a single structure. So shown here is the robot roach, which was first developed by Aaron Hoover and colleagues. And so one of the really nice things about this approach is it dramatically reduces the number of parts which we need because we can take advantage of some folding principles where we still have connected segments and we can assemble them. So that was about smart composite microstructures. Let me play a video which kind of walks through that and shows you one of the really
This is
successful
Dash.
robots built using this
Dash, or
approach.
the dynamic autonomous sprawled hexapod, is a small, high-power density, minimally actuated robot capable of high-speed running and surviving large falls. The design of Dash has been informed by the study of nature's greatest runners. From these animals, scientists have derived many models for robust, high-speed locomotion. Dash is constructed using a scaled smart composite manufacturing, or SCM, process that creates rigid cardboard beams with polymer flexure joints. Laser micromachining removes cardboard from the flexure joints in a mirrored pattern. A thin polymer is laminated between the mirrored cardboard sheets, and then the final outline cuts are made. The cardboard beams and flexure joints can be folded into complex functional elements. The scaled SCM enables an iterative design process as a result of low material costs and rapid build times. For example, Dash can be constructed in as little as 1 hour. Dash employs a single DC motor to create a sprawled alternating tripod gait, which is beneficial to dynamic stability during running. The design translates the circular output of the motor to roughly circular trajectories of the feet, much like the oar of a boat. Dash is able to run straight at speeds of 1.5 meters per second, or 15 body lengths per second. In video slowed to 1/10 real time, the dynamics of Dash are better observed. Different leg designs can easily be interchanged on the same body, and the space of possible leg designs is still being explored. Dash is able to climb over obstacles taller than its own body height, thanks in part to its high power-to-weight ratio and dynamic gait. A shape memory alloy servo motor pulls on the frame of Dash, changing the midpoint of the lateral sweep of the legs as shown. This creates a moment on the body of Dash, thereby inducing a turn. Turning currently works best at slower velocities. The cardboard beams created from the SCM process are fairly rigid when forces are directed along the length and the height, but are weak to forces directed into the face of the beam, as well as to moments about the beam. Dash is designed so that rigidity is maximized in directions which improve power transmission and gait. The compliance that remains in off-axis directions gives Dash a unique characteristic of being able to survive falls from large heights. The lack of rigidity of the cardboard to forces into the face of the beam and to moments about the beam allow the beams to bend under forces not seen during running, contorting Dash and absorbing energies without breaking. Such forces occur when falling and impacting the ground. Dash has been shown to be able to survive falls without damage and has been tested at heights of up to 28 meters onto concrete. Here in slow motion, we see Dash is thrown into the ground at 6.8 meters per second. From a second perspective, we are able to watch Dash run away undamaged. Future work on Dash includes exploring different means of turning, using different construction materials such as carbon fiber to improve power density, as well as improving all-terrain mobility.
So that was great. So since that robot, there have been a series of robots which have basically used a similar kind of idea and improved capabilities. And wanted to show you one more example of the system, so where you have basically a robot. Now it's not only tough to external settings, but it's also compliant and can conform to environmental constraints, for example. So this approach works really well, and there have been a series of legged robots which have been developed, as I mentioned, but also there have been a number of smaller robots, so mostly flying robots developed by a number of groups pioneered by the Wood Lab at Harvard. And here's an example of that. And I want to draw your attention to the size scale. So now we're not talking about a palm size scale system, but we're talking about something which is more close to the size of a penny. You can still use the same approach, to kind of build such a small system, and I'm going to show you a video of that. And one of the things I want to highlight during this video is it's really impressive that we're able to kind of like use the same approach and like fold things together and stack and laminate them. But now the process of assembly becomes a lot more challenging. And as you can see, even a dab of glue in the not the right spot can result in a system which is basically useless. So this process becomes much more challenging as we start making them smaller, and especially if you want to make hundreds of them, then this process gets even more complicated. And here's showing the process of assembly of the main chassis and the transmission, and finally attaching the wings. All right, so and these robots work well. However, as I mentioned, so it becomes really challenging to like build multiple of these systems. And so in 2012, Rob Wood's lab came up with this approach called the PC-MEMS approach, which is an extension to the SCM approach, but now taking ideas from children's pop-up books where you can still build these structures in plane, but then fold them up and take advantage of some automated folding techniques to help you simplify assembly so that you are left with a monolithic structure in the end. So here's showing the whole process. So you can integrate your actuator, sensors, and wings in a flat planar approach, and then using a set of locked and joints, so you can like pop the whole structure up, glue it in place, and then your assembled structure is released. And so you can see the device function here. And this is flying at high speed. See all intricate structures. And so the essence of this approach is essentially can be summarized in these 4 steps. So we laser machine out the parts. So we want wherever we need flexible elements, we laser machine those structures out. We de-chip them, and then we do that for every single layer. So for example, in this approach, the simplest combination is a rigid layer, adhesive layer, flexible layer, followed by an adhesive and a rigid layer stack. So 5 layers. So we do that for every single layer. We use alignment pins in a jig to kind of line everything up. And then we cure them under pressure and heat, and finally release them using the laser again. However, the main advantage compared to the previous approach is that now most of the assembly is through folding. So because we have joints in place, we can fold things up so everything remains connected, making it a lot more manageable. And this approach has been extended to make a variety of robots. So like here's a walking robot, but as you can see, the number of layers is multiplied. We have a lot more. We have 2 sublaminates, each laminate having 5 layers, and as many as 23 layers structures have been built, been shown to work really well, and the whole structure can pop up as a monolithic unit. And just to give you an idea, so like the whole robot is really capable of some impressive performance. Showing here is inverted locomotion using electroadhesion. Because these systems are really light, they can coast on the surface of water, exploiting electrowetting. We can make them sink into the surface of water and walk underwater. Also, because of the small size, these systems are capable of really highly dynamic behaviors. And we can also make them autonomous by integrating power and control on board. And so what I want to show is this origami-based approach has been really successful in a variety of cycles, ranging from palm-size scale robots all the way to penny sizes. We can even make larger systems if we have by choosing the appropriate material choice. And there's been some work from Michigan trying to build larger ant-inspired robotic systems. We can go smaller as well. Again, it becomes challenging in terms of fabrication, but there is no restriction and in terms of going small. And one of the results I want to show you is our recent work, where we basically took the 4-centimeter version of this hammer robot and shrunk it in half in every dimension to make 300 milligram robot Hammer Jr., which is a replica of the old structure, but in terms of fabrication, it was a little bit complicated. But in terms of design, we were able to basically take the same design, shrink it down. We needed to make some changes because actuation and hinge stiffness and properties like mechanical properties varies the size of scale. And so we need to make accommodations for those, but essentially the same design which worked at a larger scale was effective at a smaller scale as well. And so here you see Hammer Jr. next to an American cockroach. It's in fact smaller than, lighter than a cockroach at the moment. And you can see we're not having to compromise any design complexity. So the small robot is capable of like a variety of gaits and also runs at high speed, can turn left and right, and so on and so forth. So the take-home message, what I want to share here is this origami-based approach is really easy to scale, and I think that's one of the really nice advantages of thinking about this technique. And the other reason why I'm really excited about potentially building physical models using this approach is that we can include a number of interesting layers into this laminated substrate stacks. In the past, all the examples I've shown you are basically a very simple stack where you have a rigid structural element, a flexible element, and another rigid element. However, there are a number of groups around the country who are trying to expand that process, including us, where we're trying to integrate a number of different elements. So, for example, we can include passive or active flexures. We can include some signal transmission layers. We can include energy harvesting layers, and so on and so forth. And the overall process still remains the same. We still laser cut the individual layers, then we heat press them, fold and glue them, and we can even potentially do some pick and place electronics if we needed to, and assemble the entire structure. I'm going to show you some early work in this direction. So as I was mentioning, body laminates are typically made with just 2 elements. So you have your rigid carbon fiber and your flexible kapton. We also make actuators to power these robots the same way, but we replace one of the carbon fiber outer layers by your PZT layer, which is an active element. Then we have a passive element, which is your carbon fiber. We have some circuit elements and signal transmission layers, which are basically copper kapton kind of layers laminated with the entire structure. And so some really nice work on medical devices and sensors from Rob Wood's lab. Here's some Josh Gaffert's work, where they're beginning to incorporate signal conduction layers and also some really small electronic components. So on the right, you can see basically a spring actuated mechanism with optical sensors to be able to act as a really tiny force sensor for endoscopes and those ideas. Here's another example of trying to include fluidic layers from some of the recent work. So here, what you can see is, we are showing a fluidic actuator which is developed by Gustav Keplinger's lab based on electrohydraulic actuation technology. But by combining that with rigid layers, you can start getting interesting joint structures analogous to like spiders, where they use fluidic actuation and muscles in coordination to achieve complex actuations. Right. That's great. So the vision towards all of this work is hopefully by incorporating these complex heterogeneous materials into a single stack, we can go towards this holy grail of autonomic materials, which have been broadly defined as materials which can sense, actuate, communicate, and do computation all within. So this is what a lot of what we think as what biological exoskeletons or skin-like structures do. They're not just used there for protection, they're not just there for actuation or sensing, but they do a number of things. And by combining different elements, we can get closer to achieving many of those on the robotics side. And I'm hopeful that this direction will hopefully will lead us to being able to generate high performance, robust and autonomous physical models, which we can use to then test with high fidelity biological hypotheses. So I want to take the last few minutes and just share some resources. Whenever we talk about origami or kirigami-based design and robotics, the question is, it seems really hard, like how do we go about doing this? So I want to share some resources which have been helpful for me and my students as we're thinking about some of this. Definitely a part of this is art, and I think by doing origami, I think we get comfortable in terms of thinking about 2D to 3D designs, and that's one of the things what I've encouraged. So if you think about origami, it has a really long history, almost since the time of like the discovery or invention of paper, I want to say, and there is a number of resources online in terms of like, and wonderful artists who are still making very complex designs. So that's like some examples of traditional origami. Some more recent examples are where people are trying to replicate all kinds of structures. Here are some like really complex animals, and also, yeah. So this is some work from Robert Lang, who built a number of designs and has many books in the last like 20 years, which are really helpful in terms of getting started with origami modeling, and going to talk a little bit about this. And so a lot of this work kind of got spurred in the 90s when people started making these insect-inspired or arthropod-inspired designs, and the community just expanded because of like how complex and realistic some of these things looked. And so in the origami community, they refer to the period of like from the 90s on as like the bug wars period, where number of groups started getting involved, and there's some progress even in terms of like computational modeling and so on and so forth. And here's like a really beautiful recent example. And so rigid origami has not just been like being used for making robots, as I showed you, but it's been really popular as a means for making things like satellites, because you can collapse these structures into really small form factors. The way I showed you is a laminate process for making these origami structures, but that's not the only way. You can still use rigid structures and make living hinges or flexures in materials and then still fold them up. And the Compliant Mechanisms group at BYU has pioneered a lot of this work, and there's some really interesting results if people are interested in checking them out. And as I was mentioning, so just a brief history. So this is like the Wikipedia has really nice references and links to a lot of these. So like most of the early recorded work is from the early 1900s, and some of the formal notations and definitions for thinking about origami structures started coming up around the 50s, and like around the 80s or where like some of the first like axioms for thinking about flat folding structures, which are the basis or the fundamental basis for origami elements, were formalized. And in the 90s and beyond, there have been a number of really impressive developments, thinking about mathematical proofs, design breakthroughs, and also a number of computational tools to help make this process of design simpler. So I just want to share some general rules in terms of thinking of how we think about it. So we try to emulate even though our final robotic structures look complex, we're trying to emulate simple linkages like 4-bar linkages, the classical designs. And so when we have these like 4-bar designs, we don't want them to cross each other because we're using paper. Paper doesn't stretch, it doesn't tear, it does not like cross by, it doesn't go through itself. So that's something to be aware of, and we can make closed structures by basically cutting places, cutting in specific ways, as I've shown you here. The fundamental basis for thinking about like origami structures is crease patterns, at least in the classical sense. So you have two basic types of creases. So you have a mountain, which basically is a crease which folds up kind of like a mountain, and then you have a valley, which is a divot. So it's like you can see those as blue and red kind of traces respectively. And so some really fundamental results are, for examples like at any vertex in an origami structure, the difference between the number of mountains and valleys at that crease intersection needs to be 2. Also, another really interesting and intuitive result is if you look at like the alternating angles at a crease intersection, they sum up to 180, and there's some really interesting mathematical proofs to help think about those. But these are some ideas, and if people are interested, Eric Demian at MIT runs a wonderful course. Most of that course material is online through MIT OpenCourseWare, and I would encourage you to go check that out. Also, there's a number of like software tools which are developed to kind of make some of these designs simpler. So one of them is the tree maker from Robert Lang. I've shared a link. So this idea where you can draw basically stick figures or design trees, which help you build the entire structure. Again, talking about the folding and cutting approach. Another tool is called the origamizer. Again, so this one relies on more thinking about polyhedral structures to make your ultimate 3D model. And so you can see a design pattern which leads to this really beautiful bunny. And one of the things I wanted to point out is if you want a particular structure in 3D, then there are a number of ways of doing that. So for example, if you want to make a cube, there is like one way, which is just thinking about squares as starting blocks and then folding them up. But we can also make more complex shapes, as we show here, and realize the same 3D structure. So it leads to some interesting design kind of like trade-offs as you're like making these structures. And more specifically to help with the robotic side, so there are like two examples or two tools we use in our group. And so one set of tools is developed by Dan Arcus' lab at ASU. And so this is a software package called PuppetCAD, which helps you think about making these laminate structures on plane and how potentially you can fold them up into a 3D structure. It makes the process of design and cutting using a laser really easy and simple. And for a more computational approach, thinking more classically about like bodies. I think the Cynthia Sung's group has been doing a lot of interesting work, and specifically I wanted to highlight the Build a Bot software, which they've been working on, which makes it really easy to make some very simple structures. And here's some more resources, and I'm happy to take any questions.
You can go ahead and take any questions for all of the presenters. You can either drop it in the chat or the Q&A or raise your hand and I'll unmute you. Thank you.
Natasha, you had a bunch of questions for Jake.
Yeah, I mean, I don't want to take up the time of anyone else, of the other participants, but that was really cool too. I have questions for both of you. It was really awesome seeing some of those robots, which I know we've talked about so much. So those scintillators you talked about, I didn't know they were there in that beam path, like just the whole, that they existed until today. How do you get phase on the scintillator? Because I understand you get, you know, a scintillator can measure how much X-ray, absolute levels, but I didn't know you can get phase from a scintillator.
Yes, but I think your question is above my understanding right now.
Okay.
It's a combination of the actual material, and the ones if I'm remembering right, the ones we use are yttrium based.
Okay.
And so the key though is that they need to be really thin. You need to make them as thin as possible, and if the thinner they are, the better resolution you get from your X-rays. Why that is, I don't actually know. I just have seen it in practice. But they there's a limit to how physically thin you can actually grind them down to, and I think they come from France, I believe. But yeah, and they're easy to break and they're expensive.
As things are in these.
But what they do though, so the system as a whole, given the wavelength of the photons, the thickness of the scintillator, and the distance, is that you typically get a resolution limit of about a micron, or it's just under a micron. And you can actually, the sensors are so good now that you can actually oversample, and you're not actually adding any resolution. But yeah, if you want to do something at a smaller resolution, there's actually, so this technique doesn't actually use any X-ray optics. But there's an instrument that is downstream of the table that I was showing you, that is it's a TXM, it's a transmission X-ray microscope. And there you have optics akin to traditional microscopy, and you can get down to the tens of nanometers in resolution. But the trade-off is that the field of view is
smaller.
Is much smaller. It's on the order of 100 microns or 50 microns or something. But you also can do tomography there too.
Right.
Which we've actually never used it, and we want to do it really soon to look at very small tracheal tubes, to look at their surface textures.
I want to leave enough blank space that if someone has a question, they can ask it. But I've also noted.
Sorry, Natasha.
Yeah, go for it. Yeah.
So Jake, like you showed some really impressive videos, and so I was curious. Like I've tried to do some tomography like myself, but one of the challenges was like the post-processing part of it, and trying to. So I'm curious, like how much time it takes for you and your students to. Like how much time do you spend kind of like cleaning those up and trying to extract useful data? And are there like easy interfaces to like maybe other like modeling software, like which at least for the engineering audience, if I wanted to print something on a 3D printer, or using our approach, how easy is it to get to that point?
Well, let me start with the first part of that. So collecting data at Argonne is really fun and it's fast. So like if you want to do you want to do tomography of the leg of one of your cockroaches, right? You can do a sample in a couple minutes, okay? Whereas like your tabletop X-ray takes a couple hours. And that makes a difference because if there's any type of motion or whatever, or there's drying, whatever, you have a problem, right? So it's fast. The problem is that you create a massive amount of data. Like, there have been times where I've come away from sessions and we have 7 terabytes worth of data. And I sometimes go, I kind of wish we collected 2 samples and now that's all we have, and that's what we deal with, because the post-processing is the extremely slow part, right?
Right.
And there's commercial software like Aviso or Mira. Recently, we've been getting into Slicer, and my former postdoc, Mary Salcedo, I think has a couple of videos on this, and there are other instructional videos as well that can get you into the 3D rendering. But the 3D rendering from the data is, particularly if the data aren't super clean, meaning like you have these sharp differences between like cuticle and soft tissue or bone and soft tissue. Like bone, you can render out, segment out really fast. But for these finer differences where you have these sort of gradations in the grayscale there, sometimes the automated techniques don't work and you have to go in and trace things by hand, and you have to do it in each slice. Or sometimes you can skip slices and have it interpolate, but then sometimes you still have to go back in and clean up. So that's where it really takes person power. And it's a great way, by the way, to involve undergraduates in research, because you really need devoted time to it, right? So when anyone has kind of a large scale amount of 3D rendered data, I'm always super impressed with it, because it takes a lot of work to get to that point.
Awesome. Thanks, Jake. I think there's 2 questions. There's one question for me. I'll try to quickly answer that, and then there's one for you, Natasha, following. So I think Stacy asked the question, what is the minimum setup required to make these origami fabrication things work? Are there low-cost versions which would be accessible? Yeah, so that's a great question, Stacy. So I think, yes, there are a number of alternatives. I think in terms of like low cost, for about, so I have started I teach a class called Bio-Inspired Robotics, and we have all the students in the class. It's a graduate class where all the students build their own robots. So the setup, what we have there is we use vinyl cutters. If we have laser cutters, we can use them, but vinyl cutters are fine if the features are large, especially for like palm size scale robots. We can make those will work. And so they typically are anywhere between $100 to $300. We use a standard office laminator, the thing which you use to like, basically make certificates. So those things work fine. And just the raw materials, what we have are poster board or basic cardboard. We can use a number of different plastics. We use Duralar, polyester, films, which are used for like art filming and lamination, because they're easily available. And the trickiest part is a glue. So there are a couple of companies which are making these hot glue, which is a wax paper covered with the glue. So once you have it in a laminator, like stick it on a laminator, the glue gets transferred onto whatever material you have from the wax paper, and so it makes it really easy to release. And so I'm in the process of like preparing all of this and like making this open source. So like probably stay tuned for that in the next 6 months. I think a lot of our course material will be available, but also there are resources online from Ron Fearing's lab. So they have some designs of their old robots, which you can, and even like cut files, which you can probably start making devices right away. So that was a quick question. Hopefully that answered your question. And so I'll update my presentation to like add more links to that stuff and share it with everyone who's as interested.
Yeah. Sorry, I guess the next question's for me. I just keep wanting to say that's so cool over and over again, which I know is not very informative. But it is very cool. About the crickets, they do prefer certain leaves, so they generally pick the larger leaf, the one that is basically close to being the most optimal baffle they can. The leaf itself, its properties don't matter except in as much as that they can cut through it. So I think they wouldn't go for something like a rubber leaf. They'd go for something that they can actually chew through. And that gets sort of taken care of by the fact that they're pretty strong affinities to plants that they prefer to live on. So, yeah. Jake, you're next.
Sure. Good question from Dylan. So yeah, Mark has done a couple of aquatic things. I don't think he's published any of it. I don't know what became of it, but I do remember him putting fish in there. You can. So the issue with doing aquatic stuff is that you're now shooting through water, and the water will absorb the X-rays. And the more absorption you have, the lower your dynamic range. So yes, it is possible, but the bigger your water column, the less you're going to see. Yeah, Liz Clark actually has a paper on sea urchin arms popping off. And I think that was done in water. I haven't actually seen the paper. Are arthropods ideal for this method? Yeah, I would say yes and no. It's actually a size issue. So as you get bigger and bigger, it actually becomes harder and harder to make out what's in there if you're just doing 2D imaging. In fact, some of the bigger, we've done some big chunky insects like a Madagascan hissing cockroach, and you just see like there's all this material, and it's pretty confusing to try to make things out. So working with smaller insects actually makes things easier. But all kinds of things you can put in there. They don't have to be animals either. Yeah, by the way, you should ping Mark, tell him to send you that stuff, and then ask him. Yeah, do that.
I noticed, Jake, that you were saying that the light is a little bit broadband, and then you to make it monochromatic. So the technique that I think Kaushik mentioned in our email that I didn't touch on at all, OCT, is it uses slightly broadband laser to measure vibrations at different depths in objects. So you can use it to kind of get phase information, and you can look at phase variation to see how much something is vibrating. And you can calibrate it to picometers if you want it. So actually from that point of view, slightly a question I had was like, are you basically limited when you take images at the synchrotron by the speed of your camera?
In some sense, yes. In some sense, no. The speed though is it's not an X-ray issue, but it's just it's a light issue, right? So you need a certain amount of light to expose properly, right? And so in the early days when we were trying to do high-speed imaging there, the high-speed cameras were not good enough, and so we could make an image, but the problem is we'd have to start going up, the peak I was showing you where you're really starting to get the intensity up. So you could crank up the X-rays, but it really would limit your behavior that you're trying to do. So something like, at some point we were looking at fruit fly wing beats, and
Mhm.
we would turn the x-rays on, and you would get like a second worth of data. And so you have 150 wing beats. That's great, but the animal's instant dead. But that was a while ago. The cameras are now much better, where you can turn that down and get more real physiology with the image.
Yeah. I mean, that sounds better than what I think Holger Krapp's group told me, which is that their flies were smoke at the end of it.
Well, actually, that's something interesting I didn't bring up, which is that with this technique, typically at 32 ID, the insects are not heating up. So because most of the photons are going straight through, they're not being absorbed. There's not a significant temperature increase, and we looked at that with IR cameras. So it was actually a little surprising, but that's how it is. At 2 BM, which it's not the case. Sometimes we do get heating of the material.
Yeah. Very cool.
Material being tissue.
Yeah.
I just saw Dylan's
Sorry?
chat. I just saw Dylan's chat item. Yeah, and then he's going to ask me for a bunch of manuscripts too. Maybe you don't say that.
I'm very pleased to see like you put up things from 2005. Maybe I shouldn't make these confessions. This is not being recorded, right?
Oh yeah,
It's still being recorded. I have data from like, I don't know, 2010, 29.
well, I would call that kind
I'll
of
edit that part out, Natasha.
2010. That's pretty recent.
Yeah, that's what I think. Yeah. Kaushik, have you got any of your robots to actually interact with any actual animals yet?
No, not yet. I mean, they do interact during photo sessions, but not in an intended way. So I'm more worried about our robots kind of breaking. I think that's happened in the past. Like our spiders and cockroaches are too big, so they just go sit on the robot and then break a little hinge, and I'm like, oh no, this was like weeks worth of work.
Oh.
So no, but I think interestingly, I would love to kind of talk about that. And so there's talking to people here and Paul Shambo at Harvard, like when I was there, we were interested in trying to like basically make a spider robot which would like pretend to be a mate or a predator and like send vibratory signals, and we wanted to see how they would interact. And it'd be really cool if we can design a robot as agile as like a peacock spider, for example, and make it do all those like funky dances and try to confuse the actual animal. But it's a few years down the future, I think.
I mean, they're not difficult to fool.
That's good to know.
So we don't have the peacock spiders yet, they're Australian, but the other ones, the paradise spiders, which also do elaborate dances, you can get the males to dance just by having like a dead female there, and all you have to do is kind of wiggle her up and down, and the male's like, yes, please get on. So they're not hard to get going.
That would be great. I think, yeah, there's a lot of like interesting like communication related biological questions, and so I feel like, yeah, if we get our robots a little bit better, then I think I would love to like work with people to kind of like begin to answer those questions.
So Kash, I have a tangential question from that related to time. So everything you showed, like, it looks straightforward. I don't think I could do that in my lab. Like, how long does it take to ramp up to be able to do that? So like, if you had some idea of design, like, oh, I'll just go follow the steps you just said, but I don't think that I could. Right. So how long does it take to train to actually do this in practice? And is it just better to collaborate with someone like you who knows how to do it already?
I always welcome collaboration, so I would not say no to that. No, I think in terms of being serious, so for it takes a good amount of time. So I think if you have the designs, the fabrication process is straightforward, but there are still little things of like, you still have to like add a drop of glue. You still need to like forge certain things in place. I showed you examples from the Harvard Fly automated pop-up system. Designing that is really hard. It takes like a lot of time to just if you have like multiple folds which you want to actuate, in theory, yes, you can actuate all of them and we can create mechanisms, but there are not really any easy tools to let you do that in an automated way. We can't be like, here's my final device, build all the peripherals and give me the cutting file. So that's not there. So for us as a lab, if you probably go to like market production or something, then we can invest in those kind of resources, then become much easier. But for a grad student, I can give you my own experience. I started my lab about 2.5 years ago, and my students have been here with me for about 2 years, and we are just about getting ready to submit our first paper on the design fabrication of this thing. So, and this is like with students who came in with like interest in origami and had like surgeon-like hands. So it's taken a little bit of time in terms of like trying to iterate through the designs. And but I think like that student, after about 2 years of training, can now probably they're working on their second robot design with like significantly different changes, and we expect to get like a working version in the next like 2 to 3 months. So I think the process gets faster, and it's exponentially decreases like in terms of the time. For the first one, it's really hard, but I think if you have the expertise built up in your group, then I think making those designs can get fast. Like I would say like probably anywhere between 3 to 6 months is what I would say, from like starting fresh on a blank slate to a new design with like specific properties.
But you also have to have good hands, is that what you're saying?
We still rely on some good hands. There are tools which will help. We have a lot of like custom jigs and things which will help us. And in fact, one of the things what we are working on at the moment is there have been some groups, Daniela Russ's group at MIT and also Rob Wood has shown some examples of automated folding. So you can have some actuators, for example, either shape memory alloys or things which help you simplify the process of assembly. And with the designs I was showing you, because we can laminate these structures, we are thinking about like having a sacrificial layer, which was just to help you align and assemble things, and then we can get rid of that after the fact. And so hopefully that should make things faster, and or like it should make it easier for a novice to be able to put that together. We shouldn't need to have a surgeon every time we need to build a robot. Like that's one of the things we're trying to move away from in the lab, trying to come up with techniques to do that. It's work in progress for us.
Yeah, my understanding is origami is like that as well, right? It's really, those fold patterns get difficult to understand and make very quickly.
Yes, I think you're right. And I have struggled to kind of explain this to my students. Like when they tell me, how did you think of this design? I'm like, I'm not sure. it seemed like that would work. So I think like I haven't been able to get a really nice formal mindset, and I think like I've spent a bunch of time just like going through origami books and like making things. So in my head, I have a design library of sorts. So if I want one link system, like a classical 4-bar, this is how I would make it. Or if I want a 2-degree freedom mechanism, I have some fixed designs in mind, but always I go back to YouTube videos of like, okay, how did you get this thing to like twist in this particular way, and then like start from there. Yeah, I think like Eric Domain has a few courses on trying to like think about that, and I've tried like the first couple of things, and yeah, I think I haven't gotten on too much deeper than that. But one of my students is really excited, and I think we'll see. Come on, but yeah. I'm happy to say that my students are better than me in terms of thinking about this. So I think that's a good sign for me.
Cool.
Yeah.
Excellent, everyone. We are approaching our end time for this session. So if there's no more remaining questions, we should probably wrap it up. Really fun talks, and we really appreciate all your contributions. Thank you so much.
Thanks, Brooke.
Yeah,
This
this
was
is fun.
fun.
Yeah.
Thanks.
The
everyone.
recording will be posted on our Sims webpage and our YouTube channel. So please go watch and share widely. Thank you.
Nice seeing you, both of you. See ya.
Yeah, nice working with all of you. See you later.
Yeah, you too. See you.